Alright, some of you have already realized that finding the sequences for your operon are not as easy as just searching the NCBI in NCBI nucleotide. So you're going to need to find those and probably the amino acid sequences too for the project. Now, I'm going to help you out here a little bit and help you show you some more tools and some various ways to search NCBI and get your operons. So let's start with, since I'm doing the lac operon, let's do that. I'm going to type in lac operon, and notice I am searching on the NCBI. I'm going all databases instead of just nucleotides or just proteins, which is what we have done. We've also used BLAST on here, but I'm going to do all databases. I'm going to do lac operon, and let's see what we get. Well, what we get, if you'll notice here, is it kind of tells you where it finds in all the databases it has. Like it has books. It finds 83 books on lac operon. Notice it finds no genomes, because lac operon is not a genome, it's an operon. But it does find almost 20,000 nucleotide sequences. A bit much. We don't want to have to search through those, so this is really not a good choice. And it shows, let's see, proteins. It shows almost 30,000 proteins. Again, we don't want to search through that. But what it does show a good number of is just genes. So the lac operon, and it shows 5 genes. So let's go look at that one. And we're going to go here, and yes, it does. Lac Y and lac I. Now that's not all of them, but hey, that is at least a place to start if we're searching. And actually the better place to go and search, alright, a better way to search this is instead of just lac operon, it's better to put in lac operon K12, because you do want E. coli K12. You put in K12, and that's a pretty good search option here. And I'm going to research, or hit the search button again, I guess, and see what we come up with. And as you'll see, we've reduced the nucleotides down to 1,530, proteins are down to 94, and genes are down to 2. So let's hit genes. And sure enough, the lac Y and the lac I show up. Not perfect, but at least it's a place to begin. So I'm going to click on the lac Y. Alright, and what do we get? Well, we get what looks like an explanation of the lac Y. This says lac Y here, and we see that it's around several genes. And look, it's right after lac Z, lac Y, and lac A, and then some other gene going the other direction. So if you notice on this, if you can see it, there are arrows. And this arrow is pointing in, going this direction, going from left to right. Well, that gene, CYNX, is going on the opposite strand. In other words, going in the opposite direction on the opposite strand than the lac Y. The lac Y is going this way, the lac A, the lac Z, and the lac I. So here, I, Z, Y, A. So we have found our operon. It is here, and it's going in this particular direction. Now, what can we do about that? Well, we would like to click on this. So if I click on lac I right here, it's going to take me, well, it just focuses in on lac I. Now you have lac Z and lac Y, and you have 2 other genes that are coming before it. One going in the opposite direction here, and one going in the same. Not quite what we wanted. I kind of like the picture of this one. This one is kind of showing us the actual lac operon here all together. Now, what else can we do? Well, we can come down here, and you can notice that if you can't see it, but it says lac Y right here, and you get all of the information about the lac Y, including FASTA and a GenBank record, and I'm going to tell you about the GenBank record here in a second. But what we're looking for at this point is the FASTA, so we can click over here on FASTA, and let's go ahead and see what it looks like. And there it is. There is our FASTA for the lac Y. Now realize what we really want is the whole operon, and that does exist. The whole lac operon does exist as a file. Of course, you're not finding it in this search, which is interesting. I can assure you that it's there, but that's only the lac operon sequence combined with lac I all the way to lac A is only there because it's a commonly used operon. Most operons, you do not have the whole operon in one sequence pre-made for you. And you're going to have to go and probably find the parts for your operons. I do not know. You might search and see if you can find all of them put together, but you probably won't. Now, so let me show you what you can do. I am in, I think we hit on lac I, or did we hit on lac Y? I forgot which one we hit. Notice it doesn't tell you. It just says we are in the K12, and it gives us a location. So the location along the entire genome is at 363,179 to 361,926. So we are large to small. Is that right? 300? Yes. So this is essentially our lac, and it does start with an ATG if you notice. So that's good. I think this must be the lac Y. Let's go see, what did we click on back here? Yes, we clicked on lac Y. Alright, so this is the lac Y. But if we want to see, and we do, we need to see, there might be areas in between these. So just because Y is here to here doesn't mean that absolutely lac A starts immediately or lac Z. We need to gather lac Y and get some overlap with here to make sure we get intervening sequences. Alright, so how do we do that? Oops, clicked the wrong direction here. Let's go back. And let's go back to our sequence. Well, we can come over here. And so what I'm going to do is I'm going to grab this sequence and I'm going to change these numbers here. 361,900. Well, this is actually the smaller one. Okay, 361,363. And so I want to go backwards to get the area before it. So instead, I'm going to, let's say take 200 off. So 361, and I'm going to go change that 9 to a 7. All right, we're going to put that there. And then I'm going to take this one, I'm going to go 200 in the other direction over here. I'm going to go from 1 to 3, and what that's going to do is put 200 base pair on either side of the sequence. It's going to make this sequence larger, and we can look for some overlap with our other operon genes to make sure we got all of the intervening sequence, because there are things in between there we're interested in. So I'm going to update this, and here we go. And so it's a little larger. We're no longer starting with an ATG. That ATG should be somewhere, let's see, 70, 140. That ATG should be somewhere down here, right here. Right here is your ATG. All right, so we added this, everything before that onto our sequence. That should be 200 base pair. All right, so we added that 200 to our sequence, and then I added 200 at the end as well. And hopefully that's enough so that you can overlap between Z, Y, and A. Now there are some other things here that we can use. I've never talked about, or not much, about the GenBank file. So let's hit the GenBank file. All right, and let's look here, and the GenBank file is references. Now what you'll have is larger sequences. Now what I have when I hit the GenBank, it gave me kind of the area that I'm looking at in whatever genome I'm looking. What it's doing is that it's telling me about what the sequence that I have here. Well, since I made it extend into the other two operons, into the other two genes of the operon, I took Y and I extended that 200 base in. It tells me about those as well. So suddenly I have the amino acid sequence for lac Z, I have the amino acid sequence for lac Y, and I have the amino acid sequence for lac A. So in other words, by me extending it beyond it and then asking for the GenBank file, And this is, of course, this is called the file-up format. So it's a little easier to read because you can find things by 10. That's where the gaps are. So 10, 20, 30, 40, 50, 60. Now this is 60. So it starts at 61. This is not 70 like a regular FASTA. But then it starts at 61 and you can go find what you're wanting. It's a little easier file format for finding a location. But this sequence then, what this means is this sequence begins at LAC Z and ends at the end of LAC A. Still not enough sequence. Remember, we need to find terminators. We need to find promoters. So you still need sequence on either end of this, but you can go back and find that using this tool, all right, and extend some more, include the LAC I as well. So this is just a thing. Now, the great thing about this is there are good journal articles and references when you don't understand something about your Operon in here. Now, these are usually original references that go way back, all right, because this has to do with the structure of it and the original discovery, and it'll probably have the sequence, but there are references in here that you can use in a GenBank file. Here's one from 2013, 1998, 1997. A lot of these will give you the journal articles where they discovered these actual sequences, and so you also get the amino acid sequence. So this is a pretty good way to do it if your Operon is organized this way in the database. Now, not all of them are going to be organized as well as others. Some of these are much better researched, but I'm going to help you, give you some more tools that will help you find everything that you want or that you need for your report. This is how you get the sequence, all right, or at least one way. Let's go back, and I'm going to go back to the original, all right, and let's look at some other things. When you got to this point and we were looking at this, so here's I, Z, Y, and A, and we have this, realize you can zoom in on out and out on this. So if I zoom in all the way on this, I can get the sequence this way. All right, so you can zoom in on the sequence this way on the actual NCBI. So realize that. And you can download your FASTA from here. So if you zoom in and just want the piece you zoomed in on, you could download that. So here are some ways to do this. We will look at this later as well. There are things called tracks. You can flip the strand so that if you want the opposite strand, you can do that. You can search for a sequence in there. You can go to very similar. There are several things you can do if you want to play around and figure out how this all works. The main one you probably want to use is the zoom. So I can zoom out so that I get the LAC A, LAC Y, LAC Z. Look at them all in here. So here's LAC A, here's LAC Y, LAC Z, and LAC I. So now here they have them pointing in the other direction. All right, it reversed it here on you, but that's fine. Just keep in mind that which direction the arrows are pointing. Here they're pointing right to left. Down here they're pointing left to right. All right, and you can see that yes, this other gene is pointing in the other direction. This CYNX is pointing this way here, and it's pointing that way and that one. So just keep all of that in mind as you look at this. Now this is the actual genome itself that you're looking at. So you're at 369,892. That should be about where 368, about where the LAC I begins. So you probably, let's see, this says location for the LAC I is 367 to 80. That's right here. So that would be what you put in, you zoom into to find the early part, but you want a little bit before it because you want to find the promoter in front of it, so maybe 100 or 200 base pair before it, but that would be getting into this gene. What you really don't want is to get into this gene. You want, though, if there's an area between these, so when does this one, let's see if they, let's get off, there we go, let's get onto this gene, and it begins, does it give you the range? Yes, 368, 420, and then this one over here begins at 366, 428. 366, 428, and this one ends at 368, 420. So there are 8 bases in between them. I think that's the way I read that, if I read that right. 420, and this one begins at 428. Yes. So you want those 8 bases in between. I put in 100. Sometimes there's more distance than not. Like if you notice this one over here, there's a bigger gap in between there. So you need to make sure you grab all of the sequence between the maybe the end of this gene and the beginning of this gene, and that would get you the whole lac operon sequence. Now there are things that could go a little bit into the next gene, so maybe you take part of the next gene. A terminator sequence might be there and be part of the next gene. It's possible. The promoter, probably not. Probably that 8 bases is all you need, but you might go into the end of this CYX, especially since it's going the other direction. All right. So there are also great articles here all over this. So lots of things, and you can focus using this to find articles that take you in a particular direction. So in this assignment, there is some leeway as to how you describe the gene. I didn't tell you, I said annotate it, which is what we're primarily trying to do here, but I also describe the function, etc. And there are many ways to describe the function and how this operon works. Basically, you just need to go read some articles, and I can help you with those articles. I know a lot of them are tough, but a great thing about it is you go to some of these things and they give summaries. So you get related articles, you look at the abstract, that part of the article that gives you a summary at the beginning, you read the conclusion, and usually that's enough to get it. But a lot of these website things I'm going to show you in this video are going to give you summaries, and those summaries are great for using in your paper. And you can reference the article, but you can, read the summary, and that's usually enough. So there's all kinds of stuff to look at. I suggest you go down here and look through the rest of the stuff that they give you. Like here is a history of the discoveries. In other words, they found different variants of, say, LAC-I or LAC-Y, whichever one we are studying here. They give you genes that are related in other organisms, or that have mutations, all kinds of things that you can look at. Transcript table here. This is something new. I don't know what this does, so I'm going to click on it. LAC-Y. All right, protein length. Not sure what they're trying to do here. Just protein transcripts and proteins. They just decided to give you a output of some sort. Not sure why they put it together in this form, but it said new, so I clicked on it. Annotation. Now that sounds like it might give you some answers. If you go to the genome annotation, let's hear it explain. Nope, that's for the next thing. Let's go to this. It says it's giving us an annotation. Oh, it does give you a little bit different information. I might look at this if I were you. This might help you annotate this and find some of the. This is new. I would check that out and see if it doesn't give you some answers for where things are located. So it might make your analysis a little easier. Use whatever tools you have available here. All right. So I've shown you GenBank. I've shown you the graphics, and I've shown you the FASTA. and maybe some of the tools here. But this is all on the NCBI. The NCBI is not the only tool, all right? And to utilize, there's another tool that might also be helpful. So I'm going to type in, and this is called Regulon DB, Regulon database. Regulon is kind of like a, misspelled it, kind of like, it's like an operon, a Regulon, and so there are genes that operate together and they regulate something, and so this is the Regulon database, and operons would be included in that. So I'm going to just put that in, and we will search for that, and here is the Regulon database. So of course, what do? You put in, say, lac operon, and well, that's way too much. And it's not really, it's a lot of stuff we're not interested in. We're only interested in the lac operon. Here I see lac I, so this material here is about the lac I, so I'll click on that. So this one can give you way too much. So your idea on this database is to reduce it. And so here is our lac I, and here's our lac Z, and here's a gene that comes before the lac I going in the same direction. So you can see the arrows, and there's going in the same direction here. And so you can do that, and you can use this to get the nucleotide, the gene, all right, and here it is. And it doesn't give it quite the way we do. We were looking at lac Y. Oh, this is lac I. Okay, never mind. I was thinking that was the same as the one we did. And you can also get the amino acid sequence here, and let's see, what else can you get? Here's your amino acid sequence right there. So you can go get that here also. It's on the other one too, and we saw it on the other. Now, this gives you different information about what it can do, binding, all right, and lots of references if you need reference on a particular type of thing. If you decide, there are lots of ways you can present this project. You could present it as a history and kind of go, well, they discovered this first, and then they discovered this. You could do something like that. I've had people do that. But generally, people just present it as, here's what it does, here's how it works, here's why it's important, that sort of thing. But you are free to do this paper, this project, however your group decides. I do need the annotation, and I do need to know how it works, and I need you to describe it and make it interesting for me and people who read it. But there are other things here that might help you. Like here is AlphaFold, and so I can click on this and it'll give you the AlphaFold reference for it. Now what is AlphaFold? It's exactly what it sounds like. This is for the I protein, and you might want to use this picture, for instance. You might want to, this rotates, this is fun. So you see this is an odd looking little protein here, but this is the I protein that sticks to the O sequence. And you might want to discuss that and add that part of your project. If you're looking for something interesting to say and you need a picture or something, you could grab this and do it. Now this looks like a dot plot, and it's very, very similar. You could learn to analyze that and give that as part of the project if you find something interesting in there you want. I want you to find things that you are personally interested in to add to the project and the explanation, because your job is to explain how it works, but you're presenting, and in presenting you usually need pictures or something, and like if you need a picture of the protein, you could come here and get it. If you thought whatever this is telling you, and I am not sure exactly what this is telling you, it's not quite a dot plot. It is a dot plot, but it's of, I'm not sure, aligned residues and scored residues. I am not sure what that is telling you, but maybe you could learn and give me some explanation as maybe it could help you explain how it works or something, or how this protein functions. So I'm just showing you there is a ton of material here, material that will help you, and they give you summaries in a lot of these. So look for summaries when you're doing these, because they will help you understand things much quicker than reading one of those long, drawn-out articles. I will help you with those articles if you really need some help on one of them. So don't be afraid to ask me. I will help you weed through the article. But a lot of times you can get most of this in a summary from one of these references. And this actual website will give you summaries. So will the NCBI. So when you look for summaries of things. All right, and click on things. Let's see, let's do this one. This one might not help too much, but this is also on I. So let's go here and see what they give us. And yes, this is more like a history, I believe, but these give you FASTA sequences. Oh, it's release dates. At different times they improve the sequence, find out they made a mistake, stuff like that. And so here's an amino acid sequence that they have found, find new versions, and that's kind of a time history. Let's go to this one, and it talks about the fact here that LAC-I uses the wrong codon for a start codon. And it talks about the history of that and maybe why it does it, things like that. And so there are all kinds of cool things that you can go through here. Now this is mainly looking at the protein here, not the actual thing, but you can see that they have references here. These are the good references here. Evidence and special references. So here's one in 1973. Don't know what did they find. I do not know. You'd have to read about it. It's probably has to, but that's how you can do this. Now I'm going to go through and help you a little bit. All right. Now in this one it did not help to put K12, or maybe it would have. Maybe K12. Let's do that. Let's do LAC Operon K12, and does this improve this search? No, I don't think it does, because I think you lose information, and you wanted something about the LAC-A, and this does not. This does not. When I added K12, it actually hurt this search. So let me go back, and let me help out those of you who are working in with other operons. So let's go back and let's take a look and see what happens when we search these databases with your operon. Oops. Did I close that? No, I have a different window open. Okay, so let's try this one and see what you get. So this is the AAE XAB operon. All right, and we do get something. All right. Maybe not a lot. Let's see right here. There we go. And we get this particular gene. We can go to the next one here, and we can look, and you get information about it. You can certainly get the sequences and work from there if you're working from this database or from the other one. All right, so you've got something here. This is the transcripts. You should be able to get the transcripts for the AAEA. All right, and yeah, right here, FASTA. So there is your sequence for that genome. So that should help you find the sequence for that one. Let's try the next one here. Those of you who are working on CAD-BA. So let me grab the name here, CAD-BA. We'll copy, and we will go back over to the Regulon database. And I'll just go to the beginning just in case, like right here. And I'm going to paste that in, and we are going to search, and there you go. You can click here, CAD-BA, Reishi coli, and there you go. And you've got FASTA formats, and you should be able to find both the DNA, nucleotide here, and amino acid down here. So that will help you find those. Now the next one is, that's back to, GenBank can also be helpful. I'm going to go down here to the choline betaine. I got to get that right. Let me get this copied here. Doesn't want to grab it. There we go. Well, not too much. Well, I'm going to grab it and copy. I'll have to erase some of that. Okay. So we have to take off that Word document part there. And we'll just search. I'm going to put a space in there. I don't know if it needs it. Let's see if that makes a difference. Yes. So here you should be able to, for instance, find this. And here you've got your sequence, your nucleotide, and ooh, no amino acids. Amino acids are probably in there somewhere, but you can, yes, oh, I see amino acid. Oh, here it is. So yes, so you can get your sequence for that one. So that should not be a problem. But let's try this last one here. Now, some things are harder in some sequences than in others, and this one, oops, I didn't mean to click on that. Let me go back. Let's click here, grab this. So I copy, and I'm going to go up here, we'll clear this, and I'm going to paste. And yes, we do have ABT, TTABT. Let's see, is that the same? That is not quite the name. Yeah, it is ABT. Okay. So yes, I was thinking this one didn't work for this. But it looks like, oh, they give you some details here. Okay, so click on this, you get the nucleotide sequence for this one and this one. The hard part is going to find if there are any pieces in between there. All right, so you may have to search the entire genome of go to NCBI and search the E. coli K12 genome and find the intervening sequences between these two. So this one ends at 76 and this one begins at 24, so 976. Yeah, so there are probably about 50 base pair in between here. You'll have to go find those 50 base pair out of the entire genome, and you'll have to search for that to get that piece in between. And I can assure you that piece in between is going to be needed. So make sure you get some overlap for these. If you don't find them here, you need to find them elsewhere. In other words, this one is just kind of giving you, and the two databases may not be the same. Just because this one says it's at this particular position, the NCBI may give it at a different position, and you might want to check the two between each other and make sure that everything is lining up correctly. That'll help you also. And always check everything with the amino acid sequence. Make sure that your transcript does translate into the amino acid sequence they say it does. You do need to check that. Okay, that should do it for this one. Lots of references and summaries and stuff. That's what I'm telling you to make sure you look at when you find these. So if you go somewhere and you click on something, make note of where you find things that have to do with references and summaries, and includes regulates. Let's see what that does. Oh. Annotated projects. Link. Well, I'm just clicking on things here that look new to me. So they add functions to these things all the time. So I'm kind of interested in what they've added. I saw where they probably added something on annotation that's probably going to give you a lot of answers, which is fine. Don't mind that on this project. Okay, so please continue. Email me if you have questions. I think this is reasonably straightforward. You've got two tools. You've got our standard NCBI, and now you have Regulon DB for you to help find your sequences, and you have their tools that they produce online. And Regulon here has added a few features that I have not noticed before, so you might want to explore them. They might help you. It looks like they absolutely did. And you should have everything you need. So keep working. I I would work on figuring out how things work before you start annotating. That will help, but you need to understand how it works, just like you need to understand the LAC operon before you understand how to annotate it. You need to know how your operon operates before you start annotating. And you can find those references here on these two databases.